Welcome back, everybody. In the last video, I introduced the idea of enzymes and how they speed up chemical reactions. Today, I want to expand upon that concept and talk to you about how cells exert some control over enzymatic reactions. So let's get started. We can gain some insight by taking a closer look at what actually happens in a chemical reaction. And to use the example I gave yesterday of why a book as it ages does a slow burn, but if you put a match to it, it does a fast burn. We want to look a little bit more at what actually happens in a chemical reaction at the molecular level. For chemical reactions to occur, the reactants, the molecular reactants, have to collide with enough energy so that the bonds will be broken and new bonds will form. In normal ambient conditions, only a very few molecules have sufficient energy to cause the paper to react with oxygen. But the match speeds up enough of the oxygen in paper molecules to increase the rate of reaction. And since that reaction releases energy, it is self-sustaining until all the paper is burned up. So what an enzyme does is it causes the reaction to happen without the molecules having to increase their energy in the first place. And we'll talk a little bit about how that could happen. So the first thing I want to reemphasize from yesterday is the importance of enzyme shape. And I want to give you an idea of what enzymes look like so you can develop a deeper understanding of the importance of shape, particularly the shape of the active site. If the active site doesn't have the proper shape, the substrate can't fit into the enzyme and the reaction will not occur. The primary effects of temperature are to change the shape of the enzyme and hence to change the shape of the active site. And that's why temperature is so critical to whether an enzyme can function or not. A lot of recent work has been done to understand the relationship between protein shape and its ability to catalyze a reaction. And you can imagine computer graphics have been really important in that quest. You can use Google to find models of proteins and see their shapes, and here's a picture that you might find on Google. Here is an enzyme, and you can see it is a globular protein, and here's the substrate, and here's the active site. And in order for this enzyme to take this substrate and turn it into product, the enzyme has to fit into the active site. It's also important that you understand our model of enzymatic reactions. The substrate has to bind to the enzyme, as I just showed you. This creates the enzyme-substrate complex. The enzyme then converts the substrate into products. The products are released, and the enzyme is free to act again. And here's what that process looks like in a drawing. You can see here is the enzyme. Here are the substrates. They fit into the active site, which is here. Then the enzyme causes these two substrates to turn into these two products. They're released, and the enzyme is free to do this all over again in this cycle of catalytic reaction. How do the enzymes actually do it? Well, they have a lot of methods, and each enzyme is different. But they can orient the substrate in just the right way, so it makes it much more likely that the reaction will happen. They can stress the substrates and therefore make it more likely that the bonds will break and new bonds will form. They can provide a favorable microenvironment in which the chemical reaction will take place. And they can actually participate directly by binding with the substrate and therefore weakening bonds in the substrate and allowing the substrate to form new bonds. And that's more advanced chemistry. You're not going to need to know that for this course. And I just wanted to give you some idea of the way biochemists think about that. Since enzymes are proteins, their presence in the cell is controlled by gene regulation. So one way a cell can control enzymatic reactions is by controlling whether or not the enzymatic gene is expressed or not, the control of transcription and translation of that gene. But once the enzyme is made, cells also control the activity of enzymes by inhibiting them and activating them using other molecules. So let's see how this works. Here's a drawing. The top frame depicts a normal enzymatic reaction where you see the enzyme, its substrate, they fit together, the enzyme forms the products. But in the second frame, you can now see that here is a molecule that fits the active site but isn't converted into product. And if the binding of this second molecule is reversible, then the molecule would compete for the active site and reduce the rate of reaction. And such molecules are called competitive inhibitors because they compete for the active site. So cells could reduce the activity of an enzyme by releasing a competitive inhibitor. A second way that cells can bring about inhibition is shown in the third panel. The cell can release a molecule that can bind with the enzyme, and in so doing causes the enzyme to be unable to complete the reaction even though the substrate is still binding to it. Such molecules are called non-competitive inhibitors, and cells can release these molecules to reduce or halt the rate of the reaction. There are two other ways that cells can inhibit or activate their enzymes. First, they could release a molecule that changes the shape of the active site so that the substrate can bind, and these are called inhibitors. So they can release an inhibitor molecule, as shown in the first panel here. Alternatively, they can release a molecule that could bind to the enzyme and improve the shape of the active site and therefore increase the rate of reaction. This is called an allosteric inhibitor or an allosteric activator here, but you don't need to remember that word. Just remember that cells use these kinds of control frequently. There are other ways that cells inhibit and activate enzymes, but these are a way to more advance the course. The rate of enzymatic reactions depends, in addition, on the substrate concentration. So I want you to imagine the following situation, where there is a small amount of substrate and a relatively large amount of enzyme in the cell. If this happened, there would be very little product formed because the substrate concentration limits the rate of reaction. But what if the concentration of the substrate was now doubled? Well, you would think that the reaction would probably double because most of the enzymes are just hanging out waiting for a substrate to come along. And continued doubling of the concentration will continue to increase the rate of the reaction. But at some point, the amount of enzyme would begin to limit the rate of reaction. In other words, there would be more substrate available than there is enzyme. And so the doubling of the substrate concentration would no longer double the reaction rate. In other words, the reaction rate would start to level off. Ultimately, there is a concentration of substrate above which the rate of reaction can increase no further because all available enzymes are working as fast as they can. And we see that that's the case in this purple graph here. If we make a graph of the initial rate of reaction versus the initial substrate concentration, it'll look like this for a given amount of enzyme. It'll look like an asymptotic curve. And no matter how much more substrate you add at some point, you can't get a higher amount of product form because there just aren't enough enzymes in there. I'll leave as extra credit for you why the curve for a competitive inhibitor looks like this and why the curve for a non-competitive inhibitor looks like this. So that's it for today. What I'm hoping you understand is the importance of the shape of the active site. The model of how an enzymatic reaction occurs, starting with the enzyme and the substrate separately, to the enzyme-substrate complex, to then the product being released and the enzyme being able to do its thing again. How inhibition can occur both competitively and non-competitively, and how activation and inhibition can occur by changing the shape of the active site. In the meantime, take a break from all this studying, go outside, look at nature, ask biological questions, bring them back to class, and I'll see you soon.